So I might make a start. Thank you, everyone. Before we start, I'd like to acknowledge the custodians of the land on which we meet today, Wurundjeri people of the Kulin Nation, and pay our respects to their elders, past and present, and to any of their relatives present tonight. I'd very much like to welcome Professor John Mariadason from La Trobe. John's been working in colorectal cancer for a number of years. I've had the pleasure of collaborating with him on a number of projects. His background, he did his PhD in 1998 from University of Melbourne, undertook postdoc training in New York, set himself up there initially, achieving assistant professor status at Albert Einstein, and in 2008 moved back to Melbourne to the Ludwig to head up the oncogenic transcription laboratory. So he's currently based at the School of Cancer Medicine at La Trobe. He's a professor there, head of, again, of the oncogenic transcription laboratory at the ONJCC. And his topic today is looking at loss of differentiation in colorectal cancer and how we might target that to treat patients with metastatic disease. Welcome, John.
Okay, thanks very much, Peter. Thank you all for coming. So it's a pleasure to be at this spectacular building. So I'm based at the Austin, which is a partner of the VCCC Alliance. It's very it's a privilege to represent them and contribute to this exciting initiative. So I'm going to talk today about sort of a more recent area of research that our labs begun to undertake, which is to look at differentiation as a potential treatment or as a potential treatment for colorectal cancer. It's a relatively less well-studied aspect of the disease, but I think, as I hopefully can convince you during the during the rest of the talk, that there is significant potential for treating this aspect of colorectal cancer. So, for the benefit of those who don't study this disease or the students in the audience, colorectal cancer, as you all know, is a very common disease, but can be broken into two major subtypes based on the underlying genomic instability that gives rise to it. So the majority of colon cancers we classify as having chromosomal instability. These are cancers that gain or lose entire chromosomes or undergo a series of chromosomal rearrangements. So they're aneuploid. Instead of having the normal complement of 46 chromosomes, they can have as many as 40, as few as 40, or as many as sometimes 100 chromosomes. So they're highly aneuploid, and about 60 to 80% of colon cancers fall into this category. The other subtype, which is a smaller subset in the overall setting, is colon cancers that have what we call microsatellite instability. These tend to have the normal complement of chromosomes, 46, but their underlying characteristic is they have mutations in genes that prepare mutations, and as a consequence of loss of those repair genes, they accumulate a vast array of mutations and have what's called a hypermutated phenotype, several hundreds, if not thousands of mutations. And the sequences that are particularly prone to mutation in these cancers are microsatellites. It's simply a complex term for a repetitive sequence of DNA. This can be a mononucleotide repeat, which is simply a string of C's. It can be A's, T's, or G's, or dinucleotide repeats. And when polymerases replicate these pieces of DNA, they're very prone to slippage, and consequently these types of sequences are very prone to mutation. So these cancers can be readily distinguished simply by measuring known repeat sequences in the genome and measuring their length. So often these cancers will have a gain or a shortening in the length of standard microsatellites that are present in the genome. So that's they're the two major underlying genomic predispositions to colorectal cancers, but those predispositions result in a series of mutations that accumulate in a stepwise process, beginning in a normal cell to ultimately give rise to a full-blown tumor cell. And the pathway and the genes that drive this process are very well defined. Groups such as the Vogelstein lab that did this several decades ago, and those findings have been confirmed by more recent high-throughput genomic sequencing studies. So many of you here would know this sort of spectrum of mutations that occurs. This is on the top are the mutations that commonly occur in microsatellite stable or the chromosomal instability subset of colorectal cancers. APC mutations are what start the process off, as you can see here, resulting in adenoma formation, and you get these series of mutations that drive that cell towards greater stages of malignancy. The other pathway, the microsatellite instability pathway, these cancers have, as I said, they're initiated by mutations in DNA mismatch repair genes. That alone is not a problem, but what happens is, inevitably, when you have a mutation in those genes, you lose the ability to repair mutations in subsequent genes, and this invariably, will result in mutations in the key oncogenic and tumor suppressor genes that give rise to this cancer. And the classic spectrum of mutations in MSI is shown at the bottom there. Now, they look different. The different genes are actually affected, but if you consider them in terms of pathways, the same pathways are affected or deregulated in two types of colorectal cancer. So while BRAF mutations are common in MSI, as opposed to RAS mutations, it both result in hyperactivation of the MAP kinase signaling pathway. And there's a reason for this differential selection of different genes for mutation in the two subsets. So the TGF-beta receptor gene is mutated very frequently in MSI cancers. And if you look in the sequence, there is one of these classic microsatellites that are prone to insertion deletion mutations as a result of the lack of this mismatch repair gene. So you just get greater selection of genes with these types of microsatellites in this MSI subset, and the same for the BAX gene. So that's sort of the underlying genetics of the disease. In terms of treatment outcomes, it's important to emphasize that the disease is highly treatable, curable if detected early. That's the emphasis on early detection and screening. But in the metastatic setting, outcomes for this disease have improved over the last 20 or 30 years, as shown in this graph, where if we look back in the mid-1990s, life expectancy would be about 12 months. This has now progressed to 24, if not even longer in some studies. So there has been a steady improvement, and this is due to an increased armamentarium of chemotherapeutics and targeted agents that are available. So back in the 1990s, 5-FU, the chemotherapeutic, was pretty much the only treatment available. But subsequent to that, combination chemotherapy regimens were developed using 5-FU, where 5-FU is used in combination with drugs such as irinotecan and oxaliplatin. And more recently, a suite of biological agents targeting specific molecules or processes have resulted in further improvement in survival. So these include drugs such as bevacizumab and aflibercept, which inhibit angiogenesis. There are monoclonal antibodies that inhibit the EGF receptor, which is overexpressed in several of colorectal cancers, and also more recently a drug called regorafenib, which is a multi-kinase inhibitor that's now approved for people who've with refractory disease to other agents. So, nevertheless, despite that progress, if we look at 5-year survival rates for patients with metastatic colon cancer, they're still unacceptably low, in the range of 15%. There are a subset of patients with metastatic disease who are amenable to surgical resection, which is also important to emphasize, but nevertheless, this overall survival rate is still unsatisfactory. So we as a group, and several of us here all would, I'm sure, agree that new treatments and new treatment paradigms are really needed for improving outcomes in this disease overall. So if we think about the hallmarks of cancer that many of us will be versed with, and look at the agents that are currently used, we're probably only targeting a subset of those. So these drugs, chemotherapies, typically apoptosis-inducing agents. We have targeted agents which essentially block cell survival, and then we probably have anti-angiogenic agents. But there are a whole range of other changes that tumor cells undergo that we're not currently therapeutically exploiting, and there are various challenges associated with that. But one of those, I guess, hallmarks is tumor differentiation. Now, this doesn't necessarily fall into the Hanahan-Weinberg classic hallmarks that have been defined previously, but I was at AACR, and Hanahan-Weinberg made the comment that a hallmark can be anything you want it to be now. So I guess we're going to consider loss of tumor differentiation as a hallmark of cancer, at least for the purpose of this talk. So what is loss of differentiation? So this is what the normal colon looks like. So the cells in the normal colonic epithelium are very well organized in these glandular structures, nice circular rings of epithelial cells. Colon cancers can present with various histologies. Now, well-differentiated tumors are shown here. They have some retention of glandular structure, resembling the normal colon, but then as you can see in this series of slides, there are colon cancers that can present with progressive loss of differentiation. Okay, and in some cases, these poorly or undifferentiated cancers have almost no evidence of glandular structure, and the cells almost grow as a sheet or with a flat morphology. There are some known genomic associations with loss of differentiation. So microsatellite unstable cancers, which I introduced before, tend to be, there's an increased preponderance of poorly differentiated tumors among that subset. A lot of BRAF mutant tumors tend to be undifferentiated. And there is another phenotype, this hypermethylation phenotype called CIMP, or the CpG island methylated phenotype, which I won't discuss in too much detail, but those colon cancer, poorly differentiated cancers tend to also be enriched for that particular methylation, that deregulation of methylation. I'm not going to talk about these too much today, but colon cancers, poorly differentiated colon cancers can also present with other types of histologies, mucinous adenocarcinomas, medullary and signet ring cell carcinomas are also classified as poorly differentiated cancers. These are a little bit different to the loss of glandular structure that I'm going to focus on today. And finally, there is also this serrated type of adenomas where the glands develop in this sort of, they look like the teeth of a saw and serrated morphology. And they tend to be associated with some of the features such as BRAF mutations and microsatellite instability as well. But I'm going to focus on what causes the loss of glandular structure, which is the differentiation defects seen in the majority of colorectal cancers. Okay, so the other sort of introductory point that I want to make that's relevant to the talk is that the loss of differentiation assessed by loss of glandular structure is often also associated with loss of differentiation markers. So the stains here is for a differentiation marker called villain. It's normally expressed in the normal colonic epithelium, this brown staining, which is decorating the apical membrane of colonic epithelial cells. So that's its normal expression profile. And you can see in some differentiated colon cancers, this gene is expressed, continues to be expressed, and in that sort of membranous pattern as seen in the normal colon. And then as you lose differentiation, you tend to lose expression of this differentiation marker as well. So it is a useful tool for particularly tracking changes in differentiation when studying cell lines in vitro, which is the approach we take broadly. Okay, so why study this? And is there scope for reinducing differentiation as a therapeutic? There's a few pieces of evidence that suggest this may indeed be the case. So this is a cohort of stage 3 metastatic stable colorectal cancer. So we've taken out the microsatellite unstable subgroup to avoid bias, because they are a known good prognosis subgroup. And this study done internally corroborates a lot of other studies that have been published that differentiation grade does impact on outcome. So if you have a low grade or poorly differentiated tumor, shown in blue, outcome is significantly worse for stage-matched microsatellite, well-differentiated tumors. Okay, and if I guess if you were to see a separation of curves on a clinical trial of that nature, that would be quite exciting. So there's certainly scope for treating differentiation and improving outcomes by turning poorly differentiated tumors into well-differentiated tumors. There is some anecdotal evidence in rectal cancer that differentiation grade can impact response to chemoradiation therapy, although perhaps a little controversial. And what the observation is that well-differentiated tumors tend to be more responsive to this combination treatment. And I guess the last thing is somewhat more theoretical. And so this is the normal crypt structure of the colon, where stem cells at the base of the crypt, undifferentiated cells, give rise to progeny that move up the crypt axis, progressively differentiating as they do so. And when they get to the surface epithelium, these cells are shed into the lumen and undergo apoptosis or some other process of cell death that's a little undefined. But nevertheless, what is clear is that the differentiation program that occurs in the normal colon is a terminal process. Cells are inherently wired to, upon undergoing differentiation, to apoptose. So there is the theoretical possibility, at least, that if you were to re-induce differentiation, you would be pushing cells towards that natural program of differentiation, of apoptosis, which they're inherently programmed to do. which may perhaps explain why, in some cases, better differentiated tumors are more amenable to chemotherapy-induced apoptosis. Okay, so what I'm going to talk today, I've broken the talk into two parts. One is, first of all, the work we've done to understand the molecular basis for the loss of differentiation in colon cancer. It's surprising. There's surprisingly little is known about what are the molecular events that result in well-differentiated versus poorly differentiated tumors. We know about BRAF mutations, but we don't know much, perhaps more than that. Certainly we don't know about changes in transcription factor expression that may be driving some of this change. And the second part really is based on, well, is to ask whether there are any pharmacological approaches that we can use to reinduce differentiation. So the model system, or the workhorses in our lab that we've used to address this question is this large panel of cell lines. I'm showing you about 30 here, but we have a panel of 70 colon cancer cell lines that we routinely use for a number of our studies. Collaborating with Oliver Sieber at the WEHI, we've genomically characterized these lines for mutational changes as well as copy number changes, and reassuringly been able to show that the mutational spectrum of cell lines as well as the other genomic arrangements are very much model very accurately the changes that we see in primary colorectal cancers. So at least from a genetic point of view, they are reproducible, robust models of the disease. What we've been doing in the lab is to grow all of these lines as xenografts. It's really almost essential in order to determine the differentiation grade of each of these cell lines. So this has been a sort of an ongoing sort of side project in the lab. But you can see from the histology of these cell lines now grown as xenografts that we can clearly identify well-differentiated or moderately differentiated cell lines, shown here by the presence of glandular structure, as well as poorly differentiated cell lines which have no evidence of glandular structure. I think it's always striking to me that some of these cell lines have been growing in vitro for decades, 20, 30, 40 years. Despite that, they still retain their ability to recapture the differentiation status from which they were originally derived when grown in these in the appropriate environment. We've taken a subset of these lines and confirmed that the changes in glandular structure or differentiation status assessed by glandular structure can also be confirmed by expression of differentiation markers. So here are 4 well-differentiated or moderately differentiated cell lines, 4 poorly differentiated lines. And you can see that expression of several markers, vilin, keratins, cadherins, are expressed much more highly in the well to moderately differentiated tumors compared to the poorly differentiated subset, providing us another way of assessing differentiation change. This is staining of those tumors for keratin 20 and E-cadherin. You can see much higher expression and retention of these markers in the well-differentiated subset. So the question is what's driving the loss of differentiation? And we've always been interested as a lab in transcriptional reprogramming. Okay, is it the loss or gain perhaps of transcription factors in this subset of cancers that's driving this underlying phenotype? And the evidence for that, or I guess the theory behind that, is that when you lose differentiation, you lose a whole series of markers. Okay, so that suggests that perhaps a master regulator is being lost in the poorly differentiated subset to give rise to this change. And evidence that that is in fact the case is when we took the vilin gene, which you can see is expressed in well to moderately differentiated tumors, but not in poorly differentiated tumors. And if we put in a promoter-reporter construct of the vilin gene, we see that the reporter construct reflects endogenous expression. So the poorly differentiated subset have very little promoter activity, supporting the idea that there is a loss of transcription, one or multiple transcription factors that's causing this loss of vilin expression. So with that in mind, we took a subset of moderately and poorly differentiated cell lines and did expression profiling to try and identify transcription factors that are lost in the poorly differentiated subset. So the first thing is, as we showed before, expression of several differentiation markers are lost in the poorly differentiated tumors. Green is lost. And focus on transcription factors that are differentially expressed, or in particular those that are lost in the poorly differentiated subset. We find a series of transcription factors, several of which are known players in clonic differentiation regulation, genes such as CDX1, GATA6, But we were able to fish out and focus on this one factor called EHF. It's not lost in all cases, but it's lost in several of the poorly differentiated tumors. But we became interested in it because if we look at its normal expression profile across tissues, you can see it sort of stands out in colon. It's highly expressed in the colonic tissue. And if we look across multiple tumor types, this is data from the Broad Institute where they've profiled 1,000 cell lines from various tissues. You can see highest expressing tissue is colorectal cancer. Okay? And a number of GI sort of related cancers also retain very high expression of this transcription factor. Notice, though, draw your attention to this small subset of samples at the bottom, which are very low expression levels within the colorectal cancer subset, probably, I think, reflecting the poorly differentiated subset. So here's a transcription factor, previously not really not understood, or its role hasn't been previously studied or understood. It's expressed very highly in colon and lost in the poorly differentiated subset. So it's an interesting novel candidate for us to pursue. So the other important thing, everything to date has been in cell lines in terms of fishing this out as a candidate. But if we look across a series of primary colorectal cancers and metastatic tissues, you can see that EHF expression, blue in this case, is low. EHF expression is lost during metastatic progression, and that sort of tracks along with loss of these differentiation markers, keratin 20 and vilin, as well as this transcription, other established transcription factor, CDX1. So I've lost my animation there, but so Ian Luke in my lab, who is a PhD student, is actually co-supervised by Nick Clemens, took on this project of trying to tease out the actual function of EHF. There's nice correlations between its expression and differentiation loss, but is it actually a driver of this program, differentiation loss? So I think what Ian did was to, first of all, sorry, I've just lost my animation, but we focus here. What Ian did was to generate an EHF overexpressing cell line, HCT116 cells, which normally have very low expression of this transcription factor. It's poorly differentiated. He stably overexpressed it, and you can see you can retain this over several passages. When he first looked at differentiation marker expression, we thought we were seeing some sort of an effect. We're getting an increase in vilin and keratin 20. But when we run the PCR, including a positive control, which is a cell line that is well differentiated, you can see that the magnitude of induction of these differentiation markers is very modest compared to where we really want to be in terms of reinducing differentiation. And that's borne out here at the protein level. Here's our overexpressing cell line. Really no induction of these differentiation markers compared to the levels of a positive control. And he's done this in 2 different cell lines. And when he puts these cells back and grows them as xenografts, he unfortunately wasn't able to get any evidence of differentiation induction. He did the opposite experiment, knocked it down in a well-differentiated cell line that expresses high levels of this, but was not able to find any effect on differentiation. So we went back and remembered that EHF is lost, but so are a whole series of other transcription factors, and maybe it works in combination with one or more of these other transcription factors. So it then underwent systematically to knock EHF down with each of these other transcription factors that are lost, and it met with a lot of frustration. Combining EHF with GATA6 had no effect. Knocking down EHF with GATA6 has no effect on differentiation marker expression. Down the bottom, I won't go through it, that's just a knockdown efficiency demonstration, in which case we were able to get very good knockdown. ISX in combination with EHF makes no difference, and the same with another ETS homology factor, ELF3. Finally, though, he was able to get some success when he combined EHF with CDX1 knockdown. And here you can see when you double knock down these 2 transcription factors, now you're starting to see loss of some of these differentiation markers. And that's the same in this second cell line here. So where things started to finally get interesting is when we put these cell lines back in, grew them as xenografts, and asked what happens now at a differentiation grade. This is the normal cell line. You can see evidence of glandular structure. EHF alone has no effect. CDX1 alone does have some effects on glandular disruption. Its knockdown has some effect on gland disruption, and certainly the combination we do start to see increased evidence of these more polyformed glands, and more solid-like structures. And these are all. This is all independently scored by David Williams, an anatomical pathologist who works with us closely. So that was reassuring and exciting. Finally, after a lot of work, Ian was starting to make some progress in terms of a new combination of transcription factors that can regulate this process. And he's done some staining also, and shown that when you knock these two down, now expression levels of some of these differentiation markers, cadherin 1 and villain, are lost in the double knockdown settings. He did the opposite experiment, put both of these factors back into cells that lack them. So here's overexpression of EHF by itself and in combination with CDX1. The important point is we can overexpress these two levels of an endogenous cell line that normally expresses this transcription factor. And the same with CDX1. I think these are cells given to us by Nick, or virus particles given to us by Nick Clemens. CDX1 level can also, we can restore those to endogenous levels. And when you looked at the effect on differentiation markers, this is HCT116 cells which really don't express these. We are now seeing re-expression of these differentiation markers to levels comparable to that seen in a well-differentiated cell line, and that holds true for several differentiation markers. And this is probably the most striking result we've had in the lab for some time, where you can see you put these two transcription factors back into HCT116 cells, and you can really start to see robust re-expression of a number of these differentiation markers. This is showing the overexpression of EHF and CDX1. And I guess the thing we were really chasing is, can we start to reform gland-like structures of poorly differentiated cell lines when grown in xenografts? And again, we're not creating fully well-formed glands, but we're certainly starting to see some evidence of some sort of glandular structures. And again, the pathologist would now classify this as, we classify these as undifferentiated, and this is a poorly differentiated tumor, which is still nevertheless in the right direction of reinducing some features of differentiation. And this is staining to show that we are now starting to regain expression of some of these differentiation markers, villain and cadherin 1, and we overexpress these two. So Ian's gone on to ask, it's great to reinduce differentiation. Does that impact other facets of colon cancer biology? The important one, I guess, is cell growth, and you can see that, yeah, tumor growth is attenuated when you put these two back and reinduce differentiation. And the same with other readouts such as colony formation, which is reduced, and cell migration as well, which is reduced when you put these two transcription factors back together. He's done the opposite experiments as well by knocking down EHF and CDX1 in a well-differentiated tumor, and you can see that migration rates are enhanced, particularly by the combination, and that's invasion. Okay, so based on this, EHF, as I said, has not previously been investigated or characterized in terms of its role in colonic differentiation regulation or any aspect of colon cancer, colon biology. So we also have made a new mouse model where we've floxed this gene. We can delete this gene specifically in the tissue of choice, in this case the colon. This is work that's been done by another PhD student in the lab, Camilla Rehorst. So what we've done here is to flox out or give us the opportunity to flox out the exon 8 of the EHF gene, which contains the key DNA binding domain, the ETS binding domain. And we've crossed these mice to villain-Cree mice, which allows this gene to be deleted specifically in the colon. And you can see that we're getting very nice knockout of our gene, if you like, assessed by mRNA expression, which is significantly reduced compared to wild-type mice. However, I guess we're probably not surprised, given EHF knockdown alone in cell lines doesn't have particularly profound effects in vitro. When we characterize these mice, there's no difference in body weight when we knock out EHF in the colon. All of the major colonic lineages are retained, colonic or small intestinal lineages. Perhaps we see a slight increase in the number of goblet cells. But there's no really striking phenotype that we've been able to discern yet. We've only had these mice for probably a year and a half now. We do see, however, profound gene expression differences. This is an unsupervised separation of the gene expression profiles of our knockout and control mice. And when we look at what genes are affected, there are a number of genes involved in sort of lipid metabolism and cholesterol reabsorption. For example, these genes ABCG5 and ABCG8. These are transporters expressed in colonic cells to reabsorb cholesterol. So perhaps there is some phenotype that can be teased out in these animals if we challenge them in the appropriate way, but that's very much work that's ongoing. What is interesting, we have actually knocked the gene out in the entire animal as well, just to get a sense of what this gene does in general. So this can be done by crossing these mice to CMV Cree mice. A couple of interesting things here. There's a series of interesting phenotypes. The first is that the mice, the knockout mice, are not. There's a partial embryonic lethal phenotype. The mice are not born at the expected Mendelian ratio. Some do survive, and the ones that do survive are much leaner and in general a lot less healthy. And they, as a result, both the males and the females animals have to be culled within the males within about 20 weeks, and the females can last a bit longer. But they develop a series of pathologies that I'll show you that really require these animals to be put down early. So some of them are they develop facial warts, which we've looked at pathologically, and these are characterized by hyperkeratosis and papillomatous growth. They get infection. They're very prone to infections. Probably the major phenotype that we're seeing. These mice have preputial glands, which I wasn't aware of. We humans don't. They express the glands near the sex organs of male mice, which they use to secrete pheromones to assert dominance, I guess, in the cage. Very prone to infections in mouse colonies, but our mice in particular almost always come down with infections in these preputial glands. And they also get cataracts and corneal ulcers, and Camilla's convinced these mice, must be going blind quite quickly. So we don't know what the underlying cause of these pathologies are. A lot of them seem to be sort of immune related. Turns out the gene it's primarily expressed in epithelial tissues, but it does have expression also in some immune subsets. Dendritic cells turns out also express this gene. So we're exploring two avenues. One is that there is perhaps some generic sort of immune defect in these animals, which makes them prone to infection, or maybe that they have epithelial barrier defects that makes them in turn predisposed to developing infections. So two avenues of investigation that are ongoing. Okay, so I'm going to sort of switch gears and talk about the second aspect of the work, which is some of the pharmacological approaches we're taking to reinduce differentiation of colon cancers. And I'm going to talk about two classes of molecules that we've been interested in and working with for several years, particularly histone deacetylase inhibitors and also MAP kinase inhibitors. So firstly, the Peter Mac audience would know very much, a lot about histone deacetylase inhibitors from work by Ricky Johnston and others. These are a series of about 15 transcriptional co-repressors that are expressed ubiquitously in a number of cells, and have been established cancer targets for particularly hematological cancer. So there are a number of drugs that block histone deacetylases called HDAC inhibitors, and these drugs are already approved cancer therapeutics for hematological cancers such as cutaneous T-cell lymphoma and also multiple myeloma. Just before I talk about its role in differentiation, I just want to talk about a recent study that we've completed looking at the way these drugs actually induce apoptosis, their classic anti-cancer mechanism. And to start, I'm just going to go back to an old study in the lab where we were interested in whether these drugs could have apoptotic activity in colon cancer cells. And so we did a screen of 30 colon cancer cell lines for apoptotic response to HDAC inhibitors, and we can find very sensitive lines, shown here on the right, and also refractory lines. Okay. And a while ago, we were interested in trying to tease out what the basis for that differential response was. So we treated the 5 sensitive and resistant lines with an HDAC inhibitor for 24 hours and looked at what genes are invoked specifically in the sensitive subset. So here are 5 sensitive lines, and red indicates genes that are turned on in response to drug after 24 hours. And what we've noticed, this is now going back several years, is that the the genes that are induced are enriched for a family of genes known as immediate early response genes. These are genes that are well characterized in the literature and are induced typically upon growth factor stimulation. And these include genes such as FOS, JUN, ATF3, which I'll talk about, EGR1, EGR3, and ARC. And just an example, you can see, for example, ATF3, this is induced strongly in a sensitive cell line by 3 different HDAC inhibitors, but not in a refractory cell line. So that was all work. What we wanted to do, and this is work done by Andeli Chue, who was in the lab, he's now at Weihai, and Jansen Se, who's a PhD student, just finished. We wanted to ask, is this transcriptional program that is linked to HDAC inhibitor-induced apoptosis? We've seen this in colon. Is this true for other tumor types? Is this a generic effect of these drugs? So what was done was we screened a whole series of cancer cell lines from various tumor types for response to HDAC inhibitors. And you can see, for each tumor type, you can get resistant and sensitive cell lines, shown here for lung, for example. In general, the hematological lines tend to be more sensitive, as has been shown, consistent with the clinical activity of these agents. So we were interested in this, whether these mid-early genes are induced, and they very clearly are. As you can see here, we took 8 of our most sensitive cell lines from various tumor types, and ran time courses of following HDAC inhibitor treatment. All of the genes are strongly induced in response to HDAC inhibitor treatment, and this is shown also at the protein level. We see FOS, JUN, and ATF3 induction. But what Andeli was able to show was that of these immediate genes, it's really FOS, JUN, and ATF3 that track with apoptotic response. So we correlated the magnitude of induction of each of these genes with the magnitude of apoptosis induction across the 50 cell lines. And you can see a positive correlation. So the more FOS, JUN, and ATF3 are induced, the more likely that cell is to undergo apoptosis to these agents. So what was then done was ask, well, are any of these actually driving the apoptotic process? So each of FOS, JUN, and ATF3 were knocked down in cell lines, and we measured apoptotic response as a consequence. And it was really only knockdown of ATF3 that resulted in an attenuation of apoptosis. And we showed this across multiple cell lines. This is using multiple different siRNAs, and also in different systems. These are mouse embryonic fibroblasts that lack ATF3. You can see an attenuation of HDAC inhibitor-induced apoptosis. We've gone on, what we've been able to go on and show is the mechanism by which this occurs, and demonstrate that. So what's been shown very carefully in the literature is that HDAC inhibitors can induce changes in multiple pro and anti-apoptotic genes. This is work by Ricky and others. For example, BIM, BIC, BMF are all well known to be targets of these drugs, induced by these drugs, and have been shown to be quite important in mediating the apoptotic effects of HDAC inhibitors. So we wanted to know whether our induction of ATF3 was somehow affecting expression of these pro and anti-apoptotic genes. And when we ran correlations, so what Andeli did was to look at to look for genes that regulate apoptosis, to see which of these are correlated with ATF3 induction. In fact, what he found was a significant correlation between the magnitude of induction of ATF3 and repression of BCL-XL, shown here. So the greater to which a cell induces ATF3, the more likely it is to repress this pro-survival gene, BCL-XL. And to summarize this, basically what we were able to show was that ATF3 can actually bind to the BCL-XL promoter and repress its expression. So I think it's an important contribution to the field of how HDAC inhibitors actually mechanistically are working to the repression of these pro-survival proteins. Anyway, I'll skip that and get back on track in terms of our studies on back to differentiation, how these agents which can induce apoptosis, we've actually found to also be able to induce differentiation. So in addition, as I said, to apoptosis promoting effects, for several years, us and a number of other groups have noticed that HDAC inhibitors induce features of differentiation of colon cancer cells. So here's one example. When you treat a colon cancer cell line with an HDAC inhibitor, you see formation of these dome-like structures in monolayers. And this is an example of vectorial water transport, which is a feature of the differentiated colonic epithelium. The other things are many years ago, we were able to measure this readout called transepithelial resistance. This is a measure of tight junction function, cellular polarization, all features of differentiated epithelia. You can see when you treat with an HDAC inhibitor, this readout is strongly induced. And the other thing is HDAC inhibitors have been shown to induce several markers of differentiation. This is alkaline phosphatase, the red stain. You can see it's expressed in the differentiated compartment of the small intestine, not in the crypts. And when we treat with an HDAC inhibitor, you can see re-expression of this differentiation marker. So that's always been in the literature and sort of an interesting feature of these drugs. And sort of a thing that also distinguishes these agents from standard cytotoxic chemotherapeutic agents, which don't induce differentiation. So the other thing, though, however, we noticed was that the differentiation program that these drugs induce is incomplete. Okay, if we look at, so this is going back to the slide I showed you before. In poorly differentiated cancers, the number of differentiation markers that are lost. When we treat with an HDAC inhibitor, we see evidence that some of these markers are re-expressed. This is vilin and keratin 20, but others are sometimes induced only in some cell lines. Others are not induced at all. So the drugs do work, but the maturation program or the differentiation program that's invoked is partial. So this final study that we've been working on is to look for combination strategies to enhance the activity of HDAC inhibitors and invoke a more complete maturation program. So a few years ago, papers started to come out that inhibition of the MAP kinase signaling pathway may be an additional mechanism of reinducing differentiation. Here's one such study where BRAF inhibitors have been shown to induce features of epithelial differentiation in colon cancer cells, in BRAF mutant colon cancer cells. So we wondered, so this is Laura, a PhD student in the group, who's been looking at, asked the simple question of what if you combine an HDAC inhibitor with a MEK inhibitor? Can you invoke a more complete or more robust differentiation program? And she's got a lot of evidence to suggest that that's indeed the case. So this is staining for alkaline phosphatase. You can see that the red stain is stronger in cells treated with the combination. And she's done a series of experiments to look at other differentiation markers. This is keratin 20 coming up much stronger when cells are treated with the combination, and the same with coherent 17. She's tested this combination in xenografts in vivo. You can see that the combination does reduce in greater tumor inhibition. And when we look at these tumors to see whether there is evidence of increased differentiation, certainly what we can see from a staining point of view is that differentiation markers are coming up more robustly when you treat with the combination. And additional markers do come up. So for example, trametinib can induce features of goblet-like differentiation, which HDAC inhibitors don't. So we think here's a combination by which you can induce a more complete maturation program. The final is sort of a key one, which is evidence of glandular structures. And I don't think we're quite there yet. There are some morphological changes, but certainly this combination isn't able to really cause the classic glandular structure that we hope to try and achieve. There is clearly a need for additional work to be done. Perhaps other agents have to be incorporated in, or other strategies to continue to complete this maturation program. But it's certainly sort of a start in that direction. Just finally, in terms of the mechanism by which this occurs, we're fortunate here that there's a bit of literature in terms of transcription factors that are induced by both of these drugs. So this transcription factor CDX2, a well-known gut-inducing differentiation factor, is induced when you block the MAP kinase pathway. And it is also induced by HDAC inhibitors. And so Laura was able to show that that is indeed the case. You see this is staining for CDX2. It gets much stronger staining when treated with the combination. And then when she knocks out CDX or knocks down CDX2 with 3 different siRNAs, so you can knock down the level of CDX2, and very robustly you can block the differentiation program. So this looks like both of these drugs seem to be turning on this key transcription factor to at least induce this aspect of differentiation. Okay, so 3 summary points that hopefully summarize the talk. One is just to re-emphasize that the loss of differentiation is a key feature of colon cancer, perhaps an understudied aspect of the disease. And to make the point that this loss is accompanied by coordinate loss of multiple transcription factors. And what we've been able to show is that EHF is an important component of this, previously unappreciated transcription factor, and that it does regulate differentiation, primarily in combination with CDX1, and that the coordinate loss of these two transcription factors appears to be an important determinant of differentiation loss in this disease. And secondly, that there is potential for reinducing differentiation used pharmacologically, and one such combination that we've been working on and shown to have some efficacy in this regard is combining HDAC inhibitors with MAP kinase inhibitors. So I'll finish there and I think I've acknowledged people as I go, but there's a photo of the lab. Thank you.